In this video, we're going to talk about a type of next-generation sequencing called pyrosequencing. With incredible technological development, we've developed amazing new ways to sequence DNA faster and more efficiently, and pyrosequencing is just one of these ways. In DNA replication, when a polymerase adds a nucleotide to a growing strand of DNA, the reaction causes the release of two phosphates, and we call this a pyrophosphate. So pyrosequencing makes use of this molecule, and hence its name, pyrosequencing. Pyrosequencing works by detecting DNA in the form of light. Each time a nucleotide is incorporated to the template DNA strand, a pyrophosphate is released, which undergoes a chemical reaction and lights up. So high-tech computers detect this light and put the information together in a sequence. This process can be broken down in 4 steps: library preparation, emulsion PCR, loading, and the pyrosequencing reaction. So first, we need a strand of DNA we want to know the sequence of. Then we prepare the library. A library in biochemistry is a collection of DNA fragments that we store and clone. So to prepare it, we need to first cut the DNA into fragments. We do this using sonication or nebulization. So sonication shears DNA by exposing it to high sounds and high frequencies to agitate them and cause them to break, while nebulization shears it by forcing it through a small hole. Then the fragmented DNAs are ligated with adapters, which are short oligonucleotides. Adapters are added to the 5' and 3' ends, and we denature the double-stranded DNA by heating it so that they're all single-stranded. So we end up with a hybrid molecule you'll need to amplify using PCR, which is our next step. Emulsion PCR is just a variation of PCR that some next-generation technologies use to replicate DNA sequences. To perform emulsion PCR, we incubate the DNA with microscopic beads, the thing you see in yellow here, that are bound all around with oligos complementary to our adapters, which we stuck onto the ends of the template. So these are the oligos in blue here. This allows each single-stranded DNA to anneal to the DNA capture beads. We then dilute the mixture to ensure that each bead only has one template strand attached. Then, oil is added to the mixture. After all, it's called an emulsion because we add 2 liquids that aren't miscible. We put a lipid in an aqueous solution, and you just know that fats and water don't mix. So this creates blebs, or microvesicles, so small that they can only hold 1 bead per vesicle. So inside these drops is not only 1 bead bound to 1 template DNA, but the components you need in a typical PCR reaction. Stuff like polymerase, dNTPs, primers, and buffers. So each microvesicle is like a microreactor for PCR to happen, where the DNA undergoes clonal amplification. This way we have thousands of microreactors that cycle through PCR, simultaneously amplifying thousands of DNA on their beads. PCR makes a complementary strand of DNA, including the adapters. So in each microvesicle, we have a bead, and 1 fragment anneals to its complementary adapter site on the bead. Then, polymerase, which was added in the PCR mix, amplifies the strand from the bead, and it creates another copy. Then the original strand denatures from the bead, but the strand which was just created is connected to the bead by the sugar phosphate backbone. So it sticks around. But the strand which was dissociated floats away and anneals onto another oligo on the same bead. And the cycle repeats itself 30 to 60 more times, until we end up with several thousands of the same DNA sequence conjugated to the same bead. And this happens on every bead. PCR is important because we need many copies of the DNA. The cameras we're using just aren't sensitive enough to capture the light released when a nucleotide is incorporated onto 1 strand, but millions of lights flashing at once, well, that's a lot easier to spot. Then we break the emulsion to release the beads and deposit the beads onto a sequencing chip with tiny, tiny wells. They're like 1/3 of the diameter of a hair, so we can only fit 1 bead in a well. It's important to immobilize the DNA onto these beads because we have reagent flowing in and out of the well, so it's very easy to flush away the DNA with it, and we definitely want to avoid doing that. For the pyrosequencing reaction to take place, we'll need to use some reagents in the wells. Enzymes like sulfurylase, luciferase, and apyrase, but also other substrates needed by these enzymes like adenosine phosphosulfate and luciferin. We'll also need some polymerase and primer because we're going to be replicating some DNA. Then, once we're ready to begin, the computer begins to pump A, G, T, and C into the wells, but sequentially. Only one type of base at a time. It'll add A, then wash it out, then G, then wash it out, then T, then wash it out, then C, then wash it out, and repeat. Now inside these wells, the magic is happening. Polymerase and primer binds to the single-stranded fragment attached to the bead and begins synthesizing a complementary strand using the nucleotides pumped in by the computer. Once the computer pumps the right nucleotide complementary to the template, the polymerase will add it. And this is precisely how we're going to determine the sequence of the DNA. The polymerase is stalled until it finds the right nucleotide to incorporate into the growing strand. So, if it's stalled here, and the computer pumps in A and no light is detected, we know that it wasn't the right base. So we need to try another base. But before we do, we need to get rid of A to avoid confusion later on. And we do this using the pyrase enzyme, which is a nucleotide degrading enzyme which removes unused nucleotides. Then we pump in T, no light, no dice, so degrade that too. How about G? No light either, degrade. Then our last option, C. And it worked. Light was detected, as you can see here, from the spike. Now, why did it work? Well, we said before that when a nucleotide is added, a pyrophosphate is released. Sulfurylase enzyme then uses this pyrophosphate along with adenosine phosphosulfate and transforms it into a molecule of ATP. Then enters luciferase, the same enzyme used by fireflies to generate light. And luciferase excites using luciferin and ATP, emitting a flash of light. So this is the light that we detect. When a light flashes, it means that the nucleotide that was pumped into the chip was incorporated into the DNA strand as well. The computer will then go through another round of this. Because the DNA is loaded in a grid-like way, a camera helps keep track of the light and piece together the whole puzzle, nucleotide by nucleotide. By plotting this pattern of light intensity on the graph, the sequence of the original piece of DNA can be decoded.